So, good morning, students. We are discussing today the topic about sex determination. Sex determination is nothing but ability to distinguish one individual as male, the other individual as female, based on certain physical, physiological, ethological, and biological characteristics, so that two individuals that belong to a species can be recognized as male and female. That is what simply sex determination is. There are so many mechanisms proposed by various scientists with respect to the determination of sex. As I have mentioned here, there are 6 mechanisms. One is chromosomal mechanism, second one is genic balance mechanism, third one haplodiploidy, gynandromorphism, hormonal, environmental. So these are the different mechanisms. First one is chromosomal mechanism. We can say, though it is not proposed by any scientist in particular, we can say that this is proposed by Wilson and Stevens. Wilson and Nettie Stevens proposed this. According to this, sex is determined by chromosomes. As simple as this. Sex is determined by chromosomes. We know there are 2 types of chromosomes, autosomes and allosomes. We know that autosomes carry genes for sex determination, allosomes carry genes for autosomes carry genes for somatic characters, and allosomes carry genes for sex characters or sex determining characters. So whether or the presence or absence of these allosomes is going to determine the sex according to this theory. And according to this, based on this, organisms can be divided into 2 types, homogametic and heterogametic. Homogametic means, as the name implies, homo means same, gametes. The individual that has 2 X chromosomes produces gametes which are same, with same genotype. That means there will be 2 sets of autosomes and 2 sex chromosomes XX. So one set of autosomes plus 1X, one set of autosomes plus 1X. Thus, all the gametes are said to be similar with respect to the genotype under consideration. Hence, they are called as homogametic. The other individual is said to be carrying 2 different types of chromosomes, X and Y, along with autosomes. So when it produces gametes, what happens? One receives X, the other receives Y. So genotypically these 2 are not same because they are carrying different chromosomes, isn't it? So chromosome wise they are different, hence hetero means different. These 2 kinds of gametes are different, hence this organism is called as a heterogametic individual. Homogametic, we never, until the discovery of certain different types of chromosomal mechanisms, we always feel that female is homogametic, male is heterogametic, but that is not true. Female is also heterogametic in certain organisms, male is also homogametic in certain organisms. And thus, there are 2 different kinds of mechanisms in the first one, that is homogametic female, heterogametic male. Heterogametic male type of sex determination. So here the female produces similar kinds of gametes and the male produces different kinds of gametes. And XXXY, XX female, XY male, XX female, X0 male are homogametic female, exhibiting homogametic female and heterogametic male kind of sex determination according to this chromosomal theory. And the other one is said to be homogametic gametic male, heterogametic female. Heterogametic female. So thus there are two kinds of here. This one is male and this one is female. This one is male, this one is female. So in both there are two kinds of mechanisms. In these two, when we see, the female always produces the same kind of gametes, whereas the male, in the first one, it produces two kinds of gametes, X and Y. Thus it is heterogametic male. And in the other case, it produces two kinds of gametes where there is only one X, the other is without any X chromosome. That means we have autosomes in all gametes. Here there is one X and there is one Y in each gamete. And here, this type of male, where it receives only autosomes but no sex chromosomes, that means sex chromosomes are 0. Okay, and hence we designate it as XX female, X0 male. So here 0 refers to the absence of sex chromosome, that is X chromosome or Y chromosome, whatever it may be. It doesn't denote any special chromosome called O. There is no O chromosome. It is 0. Okay. Likewise, in case of homogametic male and heterogametic female, also here there is XX male, XY female type. That means male is homogametic, female is heterogametic. But here, in order to distinguish between these two without showing the symbols, when we don't show the symbols, we don't know whether this is homogametic or heterogametic, the XX individual or XY individual. So in order to differentiate between these two, X is replaced with Z, Y is replaced with W. Thus, we come up with two different kinds of sex determination mechanisms. One is said to be ZZZW, the other is said to be ZZZ0. So here, when we say this, male here produces same kinds of gametes and the female here produces different kinds of gametes. One with X chromosome or Z chromosome, the other with Y chromosome or W chromosome, along with autosomes in all this. In the other case, the female produces two kinds of gametes, one with Z chromosome, that means X chromosome along with autosomes, the other with only autosomes, no sex chromosomes. Thus, it is also represented as 0O. So, thus we have four kinds of homogametic and heterogametic mechanisms: XX female, XY male, XX female, X0 male, ZZ male, ZW female, ZZ male, Z0 female. So, let us see one by one. Before going to that, I would like to explain the discovery of XX chromosomes. It is in 1891 for the first time a scientist called as Hermann von Henking. We can simply call, you can say, if you can't remember the entire name, you can remember the name Henking. He found that in the firebug, which is an insect, Pyrochoris aeterus, during meiosis of the sperm inherited 11 chromosomes and the other half 12 chromosomes. That means one sperm is having 12 chromosomes, the other sperm is having 11 chromosomes only. That means here there are 5 pairs of autosomes and 1 pair of sex chromosomes. Here there are 5 pairs of autosomes and only 1 sex chromosome. So out of these 5 pairs we can get, 10 pairs. Out of these 10 pairs we can get, out of this there are 11 pairs. So here out of these 11 pairs we can get 11 here plus 1 sex chromosome. Thus there are 12. Here there are 11 pairs. Here what is happening? 11 we are getting, but only 1 X chromosome is present and one sperm receives 1 X chromosome, and in the other, here it cannot receive the other chromosome. So this is the condition seen in female. gametes, this is the condition seen in male gametes. So based on this, he suggested that there is a chromosome absent in male which is excess in female. So this extra chromosome which is single, unpaired, is called by him as X body. Is called by him as X body. We can also call this as X element. And but though he proposed that there is an X chromosome, X body, he doesn't know that what to call this as, whether it is X chromosome or not. But in 1899, McClung is another scientist who coined the term accessory chromosome for this. And after thorough research, after 3 years, he came to know that this particular X body or X element that was suggested by Henking is found to be, or called by him as accessory chromosome, is suggested to be involved in the sex of the organism. So thus, this X chromosome can be having something for the determination of sex according to McClung. And after that, there is another scientist, Edmund Wilson. While working on another insect called as Protenor, he observed that this particular male individual is said to be devoid of one X chromosome, whereas female is said to be having both X chromosomes. Okay, those both chromosomes are said to be present, and it is this that determines the sex. Fertilization of male containing X chromosome. But male is going to give rise to two kinds of sperms, X and without any X chromosome. Female is going to give rise to two gametes having X. So when these two are fertilized, it results in female individual. That means female gamete fertilized by male gamete carrying X chromosome gives rise to female individual. Female gamete fertilized by male gamete having only autosomes without any X chromosome gives rise to male individual. This is the first mechanism that was studied by Wilson with respect to X chromosome, and we can say that he is the one who discovered this, who coined this term X chromosome. Okay, but the discoverer being Henking only. And after few years, his wife, while working on Tenebrio molitor, which is commonly called as a mealworm, in 1905, she proposed that in that particular organism, in addition to X chromosome, there is another. In female, there are two X chromosomes which are same in in morphology. Whereas in male, there is an X chromosome and there is an extra chromosome which cannot be paired with the other autosomes or this X chromosome, because it is having a structure which is dissimilar with that of the X chromosome. That means morphologically it is different. So she called this, as they have used the term X for the X chromosome, she used the term Y. She denoted it with Y for this chromosome. So thus, male is found to have what we call another sex chromosome, that is Y. Thus, we can say that Nettie Stevens is the one who identified Y chromosome. Thus, 4 scientists are involved here. One is said to be Henking, who discovered it and called it as X body or X element. He discovered X chromosome, and the nature of the X chromosome, that it is involved in the determination of sex, is said to be proposed by McClung. After thorough investigation, the process of sex determination involving X chromosome is proposed by Wilson. And later, his wife, Stevens, is found to discover the Y chromosome. Thus, we came to know, in addition to the autosomes, there are sex chromosomes designated as XX in female and XY in male. Okay, coming to the first one, XX female and XY male system. It is found in Drosophila, in human beings and some other mammals, and this is commonly called as ligous type. XX female, XY male system is called as ligous type because it is also found in an insect called as Lygius tursicus. And another one is said to be Melandrium album. It's a plant. Melandrium album. When we see the morphological nature of the XY chromosomes in different organisms, in male, X is said to be long and Y is said to be short in human beings, in Homo sapiens. In Drosophila, the X is said to be normal rod shaped, whereas Y is said to be somewhat having a bend towards one end. That is how we can identify the Y chromosome. In Melandrium album, the reverse to that of the Homo sapiens is found. X is said to be small and Y is said to be long. So this is how in male, in most of the organisms, these two are said to be heteromorphic. They are non-homologous. They are not of the same size. They are not of the same shape also. Okay, by different characteristics, whether it be centromere or heterochromatin or hemizygous nature, both these two are said to be different. Okay, so according to this mechanism, female has two X chromosomes, that is, she is homogametic, and only one type of gamete, that is X, is produced. Okay, carrying X is produced, and male is said to be has one X and one Y chromosome, as it is XY male. It is heterogametic and it produces two gametes, two types of gametes, one carrying along with the autosomes X chromosome, the other carrying Y chromosome. Okay, and what the scientists say is, when the ovum with X chromosome is fertilized by sperm with X chromosome, it leads to the development of female sex. And opposite to this, when the ovum is said to be carrying X chromosome, because there is only one type of gamete, when it is fertilized by sperm carrying Y chromosome, it produces the male sex. Thus, according to the chromosomal theory of sex determination, sex is determined at the time of fertilization. At the time of fertilization. And the second thing that we have to remember in this mechanism is, in this, female is just passive in nature, because it produces same kinds of gametes. Here, the male is going to determine the sex. So, male is responsible for determining sex here. What kind of male gamete is going to fertilize the female gamete is going to determine the sex of the child. Thus, according to this theory, there are two things that you have to remember. One is, male is going to determine the sex of the child to be born, and second one is, sex is determined at the time of fertilization. And coming to the second one, XX female and X0 male type. It is found in different types of insects belonging to the order Orthoptera and Hemiptera. Okay, and like examples are grasshoppers. As Wilson first discovered this mechanism in Protinor, this is also called as Protinor type. As Wilson, we have already discussed it, discovered first time in Protinor, hence we call this also as Protinor type. And here in female it has two X chromosomes and male has one X chromosome and the other is said to be absent. So there are no pair of X chromosomes in male. Thus, female is said to be homogametic, producing all the X containing only X chromosome, and male is heterogametic, producing sperms, half of which have X chromosome plus autosomes and the other has only autosomes, no sex chromosome. So when the egg is said to be fertilized by the sperm carrying X chromosome, it will give rise to female sex. When with the egg is said to be carried by the sperm with only autosomes, no sex chromosome, it results in the development of male sex. So what is going to determine here? Here sex is determined by male only, the first thing, as in the previous case. And the second thing is, sex is determined at the time of fertilization. At the time of fertilization. And the third thing is, what you have to remember here is, the male gamete with autosomes only. Male gamete with autosomes only. That means without any sex chromosome, when participates in fertilization, it results in male sex. That is what you have to remember. Okay. Next, the third one is said to be ZZ male, ZW female system. It is seen in birds, butterflies, moths. It is seen in birds. That means even in vertebrates also this kind is said to be seen. Birds, butterflies and moths. And here coming to this, male is said to be homogametic. Male is homogametic with 2 Z chromosomes. That means 2 X chromosomes, we can call it as so. And produces only one type of gamete, that is having Z along with autosomes. That means autosomes plus Z chromosome. So both gametes produced by male are said to be only one type, whereas coming to the female is said to be heterochromatic, having one Z and one W. Thus 2 types of gametes. Female produces ovum with A plus Z and ovum with A plus W. Thus female is said to be heterochromatic. So in this case also what is happening? Sex is determined. How the sex is determined? Sex is determined at the time of fertilization only. Sex is determined at the time of fertilization. Second thing is, sex is determined by the gametes of the female, not by male. So in this mechanism, as female is heterochromatic, sex is determined by the gametes of the female but not by the male as in the other case. Okay. And the third thing you have to remember is Z is always meaning to X, referring to X, and W is always referring to Y. And coming to the last one, ZZ male and Z0 female system. It is discovered in an insect. We can call it as a moth. Moths are said to be somewhat similar like butterflies, and this is called as fumia. It is discovered in fumia. In this, female has only one X chromosome, thus it is heterochromatic, and male is said to be both. So as in the case of XX female, X0 male, this is the same case. And the only difference that we can see is nothing. The first one is said to be sex is determined at the time of fertilization, and here the female is going to determine the sex. And what you have to remember is the female without any sex chromosome, carrying only autosome, when is involved in reproduction, in fertilization, it results in what we call the female individual. So that is how the sex, according to the chromosomal theory of sex determination, is determined by sex chromosomes. Hence we call this as chromosomal mechanism or chromosomal theory of sex determination. There are 4 scientists involved in this: Hanking, Macklin, Wilson and Stevens. But we attribute the discovery of sex chromosomes to Hanking, who called it as X body or X element. The sex determining nature is said to be given by Macklin, and the theory as it is proposed by Wilson and Stevens, where Stevens is the one who discovered Y chromosome. And the second theory is said to be Haplodiploidy. So the name implies here, one sex is said to be haploid, the other is sex is said to be diploid. This is as simple as this. So when only one set of chromosomes is present, that results in male sex. When 2 sets of chromosomes are present, that results in female sex. And this can be seen in the order Hymenoptera, where honeybees are present, and also we can see this in ants and termites. So because it is first discovered in honeybees, this is also called as Hymenopteran type of sex determination. Because this is first discovered in honeybees, which belong to the order Hymenoptera of class and sector of phylum Arthropoda. It is also called as Hymenopteran type of sex determination. So what happens is the females are found to be of 2 types, queen and workers. Among workers, the female which feeds on royal jelly released by the worker bees develops into the queen. And the queen is fertile because there is something in the royal jelly that is fed upon that makes it fertile. Whereas all the other females, that means workers, which do not feed on royal jelly, they remain as sterile females. And coming to the males, males are said to be called as drones. Okay, and these males are said to be having only one set of chromosomes. Okay, now what happens is, scientists found that females have two sets of chromosomes, one set with 16 plus other set with 16. Thus, the female has 32 chromosomes. N plus N is equal to 2N. Thus, female is diploid, whereas male has only one set of chromosomes, that is 16. Thus, male is said to be haploid. So what happens actually here is, there is a male individual, there is a female individual. So we know during the formation of gametes, what kind of division takes place? Meiosis takes place. So female is having 32 chromosomes, and due to meiosis, meiosis 1 and meiosis 2, it results in ultimately two kinds of gametes, such as two gametes, one with 16 chromosomes, the other with 16 chromosomes. Thus, there are two female gametes produced with haploid set of chromosomes. Okay. Next, coming to the male, scientists say that male is going to produce sperms with 16 chromosomes, because it has 16 chromosomes. If meiosis takes place, what happens here? They must be equally distributed between the sperms, having only 8 chromosomes. But here, scientists also found a new phenomenon where during gamete formation in male, in honeybees, only mitosis takes place, and it results in gametes carrying 16 chromosomes. Now, when this male gamete fertilizes the female gamete, there are 32 chromosomes, and it results in a female individual. And we can see an unfertilized egg here. So this unfertilized eggs develop parthenogenetically. It is a mechanism of asexual reproduction. The unfertilized eggs develop parthenogenetically, and it develops into a male individual with 16 chromosomes. Okay. So here, this male individual thus is not having any father, only mother is said to be present. So when it is derived, when an individual is derived from a single sex, this is nothing but a asexual mechanism. Whereas the female is always produced by a sexual reproductive mechanism with both father as well as mother. This is what we call it as haplodiploidy type of sex determination. And the third theory is said to be Genic balance theory. It is proposed by C.B. Bridges. Genic balance theory is proposed by C.B. Bridges. So here, C.B. Bridges, in one of the experiments this is also proposed in Drosophila melanogaster. So for Drosophila melanogaster, we attribute different kinds of sex determination. Okay, but this is the one that was discovered after that chromosomal theory of sex determination says that male is having XY and female is having XX. And they have come up with a different solution there. But C.B. Bridges turned the upside down with this Genic balance theory as explained by the experiments done on Drosophila melanogaster. So in one of the generations, he found a triploid female. So let me represent the autosomes. Let me represent the autosomes with A. So here A refers to one set of autosomes. That means if Drosophila is having 4 pairs of chromosomes, okay, these 2 are XX chromosomes. So these 3 pairs, 6. 3 pairs are 6 are autosomes. And I am representing only one set, that means this one, and this one. These 3 belong to one autosomal set. So these 3 form into one set, and I am representing this with capital A. And coming to the other one, the other set. This one is said to be representing the second set. That is also A, another set. So with respect to autosomes, there are 2 sets. we can show this as A plus A or 2A. AA or 2A. So when we represent only one A, that means it's a haploid female or haploid male, whatever it may be, it is haploid. When we represent 2, it is diploid. When we represent 3, it is triploid. When we represent 4 A's here, it is tetraploid. Each A here represents one set of chromosomes. It is not single chromosome. Okay? And when we represent X, it refers to not one set of X chromosomes, one chromosome only, one sex chromosome only. So that is the difference we have to observe here. So whenever there in the slide or next slide, if you are going to see A and X, A refers to one set of autosomes, X refers to single X chromosome. Okay. Now in one of the generations, he observed a triploid female. Triploid female means 3 autosomes and 3 sex chromosomes, XXX, and he crossed it with a diploid male, A plus XY, A plus XY. And you can observe here, this triploid female can produce gametes of different kinds. Gametes cannot be equally distributed here because there are 3. If there are 4 autosomal sets, 2 sets come into one and 2 sets can come into the other. So here it can produce AA plus A, that means 2 autosomal sets into one gamete and 1 autosomal set into the other gamete. Likewise, XX plus X. So this is one kind of gamete produced. And the other kind being AA plus X and A plus XX. So these different kinds of gametes are produced by the female. Coming to the male, male produces only 2 kinds of gametes. A plus X, A refers to autosomal set, and A plus Y. Now what happens? If this gamete is fertilized by this one, there will be triple A, triple X. There will be triploid female. If this gamete is fertilized by A plus X1, if it is fertilized by this one, we will have 2A plus 2X. That means it will be a diploid female. If this gamete is fertilized, AA plus X is fertilized by A plus X, triple A plus double X, triploid female will be produced, triploid female. If this one is said to be fertilized, A plus XX with A plus X, there is AA plus triple X. So far we have received AA plus XX or AAA plus XXX. We call them as diploid female and triploid female. But when he observed this is found to be not having any female characteristics. It is something special. And he observed it morphologically and can come up with another term. And likewise, when we see the fertilization of this one, AA plus XS with A plus Y, it results in AA plus XY, XXY. So here also, we know that male is said to be AA plus XY, isn't it? AA plus XY. But here we got a male with 3 autosomal sets, 2 X chromosomes and 1 Y chromosome. And he has to observe morphologically and determine what sex actually it is. Likewise, AA double X, A plus X, when he combines with A plus Y, 2A plus XY. That means a male, a diploid male is to be produced. When AA plus X is fertilizing A plus Y, then what happens? Triple A, AAA plus XY individual is produced. So he cannot call this as male because male is said to be having AA plus XY. Here there is an extra autosomal set. So again he has to determine their sex by going for morphological determination. Okay, and he observed different kinds of male and female sex determining mechanisms in this. And he has come up with what we call a sex index to determine the sex. So this sex index is nothing but X by A ratio. Sex index is nothing but X by A ratio, where X refers to number of X chromosomes. Okay, don't forget, write it down anywhere. X refers to in X by ratio, which is called as sex index. According to C.B. Bridges, in Drosophila melanogaster, sex is determined by the X by A ratio. X refers to number of X chromosomes. A refers to number of sets of autosomes, not number of autosomes, number of sets of autosomes. So that is what you have to remember. So let us see what this ratio actually is. We'll see the first case. That is, there are two autosomal sets. There are two autosomal sets, AA plus XX. So how many are there here? Two autosomal sets and two sex chromosomes. Then the X by A ratio will be 2 by 2, that is equal to 1. So according to him, when the sex index results in 1.0, the value of X by A is 1.0, she always will be a female. She always will be a female. So it is there are two sets of autosomes and two sets of chromosomes. We call this as a diploid female. Okay, let's see another case. Triple A plus triple X. Here the number of autosomes are said to be 3 autosomal sets, and the number of X chromosomes are 3. That is also 1. But here there are 3 sets. We call this female as triploid female. If we take 4 AA plus XX, that means there are 4 X chromosomes and 4 sets of autosomes. Still the value is only 1. Thus, in all these cases, it can be called as a female, which is tetraploid. Thus, what you have to simply remember is the female is having a sex index of 1.0. A diploid female 2 by 2, triploid female 3 by 3, tetraploid female 4 by 4, and so on. All are said to be having the same sex index, that is 1.0. Okay. Next, coming to the female, it is having 2A plus XY, AA plus XY, isn't it? And here there are two autosomal sets and only one X chromosome. We are not. C.B. Bridges does not include Y chromosome in sex index. Why? We will see later. For the time being, let me explain this slide. So there are only one X chromosome, and there are two autosomal sets, AA plus XY. So 1 by 2, and this is 0.5. This is 0.5. So if there are 4 autosomes plus XXY, then what happens? Number of sex chromosomes is 2, number of autosomal sets is 4. 1 by 2, again 0.5. So when the sex index is 0.5, it is a normal male. And the sex is, when the sex index is 0.5, it is a normal male. So in both these cases, 1 by 2 and 2 by 4, where it gives rise to a sex index of 0.5, it is always male. If that is the case, there is a male with 2 autosomes and only one X chromosome received, the other is without any sex chromosome. How this happens? We know a phenomenon called as non-disjunction. While critical review of Mendel, I have explained it. Non-disjunction means failure of separation of homologous chromosomes. There are 2 X chromosomes in female. They have to go one into one gamete, the other into other gamete. So if they fail to do so and come to one gamete only, the other gamete receives only autosomes but no X chromosome. If that is involved in fertilization, it results in this type of genotype, AA plus XY, X0. So in AA plus X0, what is happening here? The sex index, let us see. Number of X chromosomes is 1 and number of autosomal sets is 2. And sex index is 0.5 only. It is 0.5. But scientists say that upon morphological observation and fertilization, whether it is taking place or not, reproduction, they found that this male, though it is having a sex index of 0.5, it is sterile. Okay, male with 0.5 without Y chromosome is sterile. That means male with Y chromosome only is fertile. Based on this, C.B. Bridges concluded that Y chromosome is not responsible for the male determination, sex determination of male. Rather, it is responsible for the fertility of the male. Then what about the male sex determining genes? Here in this case, when you observe this, here there are two autosomes and one X only. Okay, so when there are two autosomes and X, no Y chromosome, we can say that it must be female. Still it is male. X is responsible for female sex. Y is not responsible for male sex, because even if it is X0, it is male, though it is sterile. Then scientists concluded based on this that autosomes carry genes for male sex determination. So with this experiment, they have come to a conclusion that in Drosophila melanogaster, autosomes carry genes for male sex determination. X chromosome carries genes for female sex determination, and Y chromosome does not carry any genes for sex determination of the male, but it carries 4 fertility genes. It carries 4 fertility genes, thus it is required for fertility of the individual. If it is absent, still too it is male. Only when one X is present, autosomes are present, it is male, because autosomes carry genes for male sex determination. So, for determining whether it is male or female, here autosomes and X chromosomes are sufficient. For determining whether it is fertile male or not, Y chromosome is required. And let's see the other case. In another case, the sex ratio is said to be greater than 1. It is greater than 1. You see the first two cases, it is 1.5, and this case it is 1.3. So what is the sex index in the first case is 3/2. What does it mean? 3 means 3 X chromosomes. 3 means 3 X chromosomes, and 2 means 2 sets of autosomes. That means AA plus XXY. Here the ratio is 3/2. And the value is 1.5. That means here this female is said to be having more number of X chromosomes. So that means when compared with autosomal sets, the more number of X chromosomes means more female sex determining genes. More female sex determining genes. That means in this the female characteristics are overexpressed, might be, and hence scientists call this individual as super female, which is also called as meta female. And coming to the male, there is a super male also. The value C 0.33. That means it is less than 0.5. If it is 0.5, it is male. Whether it be fertile or sterile, that is not the case here. We are dealing with only the sex index. So if the sex index is less than 0.5, that means 0.33. How it got? The sex index is 1/3, where 1 refers to 1 X chromosome, 3 refers to 3 autosomal sets. That means number of genes for female sex are less, number of genes for male sex are said to be more present on autosomes. Thus, it is overexpressed, the male is overexpressed, and it becomes a super male or meta male. And coming finally to the last one, that is 3/4 or 2/3. In both the cases, what is happening here is there are 3 X chromosomes, XXX, plus 4 autosomes. AA. That means female sex determining genes are also more in number. Male sex determining genes are also more in number. They cannot outnumber each other. They cannot outnumber each other. That means it is neither male nor female. And this becomes a sterile individual with both male and female characteristics called as intersex. And here also 2 by 3. 2 X chromosomes plus 3 autosomal sets. And here also neither male sex determining genes are more, female sex determining genes are more. They cannot outnumber each other. And this also results in intersex. So when you observe an index between 1 and 0.5, an index between 1 and 0.5, it is always a intersex. Okay, so thus we can simply say that 0.5 is male, less than 0.5 is super male, 1.0 is female, greater than 1 super female, and a value between these two, between 0.5 to 1, results in intersex. So based on this complete experiment, in this experiment, C.B. Bridges found that there are 2 kinds of chromosomes involved in sex determination. One is said to be autosomes, the other is said to be X chromosome, and in addition to these, we also have Y chromosome. According to C.B. Bridges, there is no gene for male sex determination on Y chromosome. That means genes for male sex determination are absent on Y chromosome. Then where they are present? They are present on autosomes. So genes for male sex determination are present on autosomes. And genes for female sex determination are present on X chromosome. And Y chromosome, though genes for male sex are said to be absent, it carries genes for fertility. So these 4 things you have to remember. Autosomes carry male sex determining genes. X chromosomes carry female sex determining genes. Y chromosome does not carry any sex determining gene, whereas it carries genes for fertility of male individual. So based on this, they concluded that they compared it with the balance. Okay? So if something more weight is placed here, the balance will move in this direction. Okay? If something more placed here, the balance will move in this direction. Isn't it? So based on the weight only. According to him, if the autosomes are more and X chromosomes are less, what is happening here? The male sex determining genes are more in number, so it results in a male individual. If X chromosomes are more, autosomes are less, the balance tilts towards female type, and this results in a female. So hence he called this as Genic balance theory. Why it is called as Genic balance theory? Because the genes for sex determination present on autosomes, if they are more in number due to the presence of more autosomes, it results in male sex individual. If the genes for female sex determination outnumber the genes for male sex determination being present on the X chromosome, the female sex determining genes, if they are more in number, then what happens? It results in a female individual. When both almost could not outnumber each other, the balance is somewhat same. In that case, it results in an intersex. If more male sex determining genes are present, it results in a super male. If more female sex determining genes are, that means more X chromosomes and less autosomes, then it results in a super female. So according to CB bridges in Drosophila melanogaster, sex is determined by the delicate balance between the sex determining genes, male sex determining genes present on autosomes and female sex determining genes present on X chromosomes. And coming to the monogenic sex determination, the fourth one I did not mention here. The expression of sex is influenced here by only one gene. In Drosophila again, they found a gene, TRA gene. TRA for transformer. This is present on autosomes and this is playing an important role in expression, important role in sex expression. So the transformer gene is found to be recessive and does have capital TRA or small TRA on either sex. That means both dominant, that means homozygous, both recessive, homozygous, one dominant and one recessive, heterozygous. In homozygous condition, small TRA by small TRA, this gene transforms the normal diploid females into sterile males. This is what is observed by the scientists. So thus, here only one expression of sex is influenced by a single gene, and such a type of sex determination is called as monogenic sex determination. And the same is found in human beings also, SRY. Okay, the gene is probably found to be present on autosome and transforms the normal male XY, if this is present, into female. Thus, it is also responsible for monogenic type of sex determination. And coming to, there are three other theories. One is environmental sex determination, the other is hormonal sex determination, and finally sex mosaics. And these three, as I have not enough time to deal with, I'll deal with them in the evening class. Okay, so evening class will be at around 8:30, and probably we'll be having the class for half an hour or more, and remaining 15 minutes we can discuss about the entire topic. Whatever questions you have now, please go through the topic till now we have discussed, and whatever the doubts you have, they can be clarified in the evening class. So we'll finish the sex determination class today itself. Okay, in the evening class will be sure, it will be around 8:30. So any doubt, please ask me, otherwise we'll close it. Thank you.